My poster is titled The Impact of Elevated Temperature and Carbon Dioxide on Growth, Physiological and Immune Responses of the Common Hourglass Tree Frog. Introduction. As amphibian development takes place in aquatic environments, they are one of the most vulnerable groups to climate change. In this report, the primary aim was to investigate the chronic physiological responses seen in early larval to adult stages of the common hourglass tree frog. It explores the development of these tadpoles under specific circumstances, such as raising the temperature and adding more carbon dioxide to their environment. This lowers the pH. It shows how the metabolism and function of the cells in the animals are affected and what this means for their growth and survival in the future. The methods used. The common hourglass tree frog was chosen as the species are more sensitive to environmental fluctuations, especially in temperature. The egg clutches were collected from home gardens in Kandy, Sri Lanka. They were placed in 5-litre glass aquariums. Once the tadpoles reached the free swimming and free feeding stage, they were randomly assigned to one of the 5-litre treatment tanks. There were 15 tadpoles in each tank. The experimental setup consisted of 4 treatments, 2 elevated temperature treatments, 1 elevated carbon dioxide treatment, and a control treatment at ambient temperature and carbon dioxide. In the elevated temperature treatments, the water temperature was maintained at 32 degrees Celsius, referred to as ETEM32, and 34 degrees Celsius, referred to as ETEM34, to represent the predicted temperature increase of 2 degrees and 4 degrees by 2100. In the elevated carbon dioxide treatment, referred to as ECO2, carbon dioxide was bubbled into each assigned tank until the pH of the water was within the range of 5.5 to 5.6. The control treatment was kept at 29 degrees Celsius, plus minus 1 degree, and the pH was kept at 7. The experiment was repeated 3 times. The results. In ECO2, the only deformity found was beak snouts, while in ETEM32, tail kinks, edema, beak snouts, and pale pigmentation were observed. ETEM32 had the highest percentage of deformities. As the tadpoles in ETEM34 died at an early stage, the deformities could not be identified. Figure 1 shows the different types of deformities compared to normal tadpoles. A through to D are the deformities, and E to F are normal tadpoles. Figure 2 shows the difference in growth of the tadpoles between each treatment. M is when they reach metamorphosis, and D is when they died if they did. In the graphs, you can see the elevated amounts of CO2 sped up phenological progression of the tadpoles through the Gosling stages. This resulted in lower body size at metamorphosis. You can also see that the development was seriously delayed and the growth of the tadpoles was reduced in both the elevated temperature treatments. In ETEM34, 100% mortality was observed before the tadpoles reached metamorphosis, while in ETEM32, all tadpoles died after metamorphosis. In Figure 3, the white blood cell count is shown. The WBC count is significantly below that in the control in both ECO2 and ETEM32 treatments. Means of the WBC counts with the same letter are not significantly different, at p equals 0.05. Conclusion and future directions. The vulnerability of amphibians is being increased due to climate change, and elevated temperatures and carbon dioxide levels are significant features of this issue. The experiments have shown that climate change can cause problems with animals' motility, immunity, growth, body size, and can create physical deformities. This study has shown that it's crucial that climate change is stopped before too much damage is done and we lose these important animals.